Okay, guys, so what I'm going to do is I'm going to go through this and answer the questions pretty quickly just to make sure that we're on the same page. Okay, so the first question says, what are three differences between RNA and DNA? And so this is just the basic what we covered at the very beginning of this chapter. You've got single strands for RNA. The sugar backbone in RNA is ribose rather than deoxyribose in DNA, and it has uracil instead of thymine. Okay, which type of RNA contains instructions? It's mRNA. The RNA that makes up ribosomes is rRNA, and the RNA that transfers each amino acid is tRNA. All right. During transcription, what is produced? RNA is being produced. You're going from your master plan to your blueprint. Your blueprint is your RNA. Where is it produced? In the nucleus. And so a great question that was asked in one of my periods by someone was, okay, well, in prokaryotes, where would this happen? So obviously prokaryotes don't have a nucleus because they don't have any organelles, and so it would just happen in the cytoplasm. Okay. Translation, what's being made? Well, you're stringing amino acids together to make a polypeptide chain, and once that polypeptide chain, twists and contorts a little bit into a 3D shape, that makes a protein, and this is done in the cytoplasm. So the mRNA has left. Everything with the ribosome is happening in the cytoplasm. Next, what region of DNA tells the RNA polymerase where it can start transcribing? That's the promoter region, okay, the ta box, TATA. During RNA editing, what is cut out, what is kept? So introns are what are cut out, exons are what are kept. The long chain of amino acids is called the polypeptide chain. So you have a bunch of amino acids together, they're held together by a peptide bond. And if you have a lot of those amino acids that are held together by a peptide bond, you have a polypeptide chain. 10, the genetic code that reads 3 letters at a time, that's a codon. If we're looking at the codon but on tRNA, we are talking about the anticodon. And the peptide bond connects amino acids together. Guys, when I saw a lot of people confusing peptide bond with hydrogen bond. A hydrogen bond is what connects your bases together. So if you have like C and G in DNA, the C and G are connected loosely by a hydrogen bond. So hydrogen bonds are pretty weak, so that enables the DNA to be broken apart. Central dogma bio tells you that whenever you're making a protein, you go from DNA to RNA to protein, no matter what organism you're dealing with. Okay, these are the different types of mutations. You've got chromosomal and point. You've got 4 different chromosomals, and we'll look at that. For point, you've got substitution, deletion, insertion. Insertion and deletion are also called frameshift mutations because they shift the reading frame, which is normally you have 3. If you get rid of one or add one, that shifts the frame, so messes up all of the amino acids afterwards. Substitution normally either has no effect or it'll just mess up one amino acid. All right. So for 15A, I've got A, B, C, D, E, F, G, H, I. I got rid of J. So this is point. You're just dealing with one base. Okay, all my letters represent bases, and it's a deletion. Here, guys, I forgot to put the J there. I'm sorry. I have A, B, C, D, E, F, G. And if you notice, the H, I, J, K, L are in order, but they're flipped, that whole region. So it's chromosomal because it's a big chunk, and I've inverted it. Okay. For C, I've got A, B, C, D, P, E, F, G, H, I, J, K, L, M, N. So the P didn't get rid of anything. It was just inserted in there. So it's point and it's insertion. For here, A, B, C, blank, E, F. So the O was substituted for the D. So it's point and it's substitution. A, B, C, D, E, F, G, E, F, G, H, I. So that's a repetition. So that's a duplication. It's chromosomal because it's a big chunk. I have a lot missing here, so that's chromosomal and deletion. Here I've got X, Y, Z, which has no place there, so it's chromosomal because it's a big chunk, it's a bunch of letters, and it's translocation because the X, Y, Z came from a totally different chromosome. Something that causes cancer, which can be physical or chemical, is a mutagen. When I'm talking about operons, guys, I'm talking about prokaryotic gene regulation. And so the trip operon, what happens, if I have trip, that causes the repressor to be turned on. The repressor just binds to the operator, and all it does is it prevents transcription from happening. So if the repressor is turned on, no transcription is going to happen, and then therefore no translation. The reason why, this gene makes TRP. So if I have plenty of TRP, why would I want to make more TRP? And so that's what it's doing. It's saying, whoa, 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 we got plenty of TRP. We don't need to make any more. Therefore, the repressor is going to be turned on. It's going to block transcription. Okay. For the lac operon, the gene actually codes for an enzyme that breaks up lactose. So there's no reason to have this gene working, right, to be making RNA from the DNA and proteins from the RNA if you don't have lac, because then you're not needing to break anything up. And so when lac is present, the repressor is actually turned off, it's removed, so then your transcription can happen. If you don't have lac, there's no reason to have the protein made, which will break down lactose, so the repressor remains on the DNA, so nothing's being made. Because the only reason why this should be made, it's making an enzyme that then breaks down lactose. So why break down lactose if you don't have lactose? So they're the exact opposite. Make sure that you understand them, because I can easily ask a couple questions about it. In eukaryotes, guys, we have what are called transcription factors. Transcription factors, they're very, very complicated, but for your intents and purposes, in order for RNA polymerase to start at the promoter region, the RNA polymerase needs to be brought there by transcription factors. They help unfold stuff. They help just in the whole setup of getting transcription to happen. So transcription factors determine whether or not eukaryotic genes are going to be expressed. Okay. How does RNA interference serve as a form of genetic regulation? Well, what RNA interference does, you have this hairpin loop of RNA that's formed, and then it's spliced up, and then you have two together. They end up splitting apart, and you have just this microRNA. It's a little chunk, like 12 nucleotides long, of RNA. And what it does is it joins with proteins to make what's called a silencing complex. That silencing complex then goes in, and whatever that little chunk of RNA is complementary to as far as mRNA is concerned, it'll go bind to that, and the silencing complex actually breaks apart the mRNA. So the whole point of this is it's not stopping the mRNA from being formed, it's actually stopping proteins from being made by destroying the mRNA. And that's being used in labs a lot to be able to stop certain proteins from being made. All right, I'm going to go through multiple choice pretty quickly. I don't think it's too bad. 20 is B. You've got 2 strands for DNA, 1 for RNA. What is the form of RNA that carries the code? That's mRNA. Which of the following carries the amino acids? That's tRNA. This was review from last chapter. I don't know why I put it in there. You're going from 5 to 3' when you're having DNA replication. The synthesis of RNA strand from a DNA template is called transcription. Translation is when you go from RNA then to protein. 25 is C. You do not have the ribosome reading the mRNA in the nucleus. You have everything else happening in the nucleus. This happens in the cytoplasm. 26, the portion of mRNA molecule that is actually translated. So that would be C, the exons, those are what are left over. 27, RNA does have a sugar phosphate backbone. So this is not true. All the rest are differences. This is not a difference, if that makes sense. All right, so I want to find out what's complementary to this strands, and so that's C. You've got U, A, C, G, C, A. Where in the cell does translation occur? In the cytoplasm. Okay. For here, we've got 3 different options. You've got 3 different places, 4 different letters that can happen in each of them. So what you do is you say 4 times 4 times 4. Those are all the different possibilities. So there's 64 different codons, but as we talked about, there are only 20 different amino acids. So obviously some codons are going to code for the same amino acid. So 64 different codon options, only 20 different amino acid options. 31, permanent change, that's mutation. Here, this one's kind of funky, guys. So my anticodon is GCC. Okay, remember the table that I gave you, you need to use your mRNA codon. So therefore, I need to look at what is complementary to this. So for the mRNA, it would be CGG. Okay, so don't use the anticodon to find this. You need to first find the mRNA, which would be CGG, and therefore it would be the amino acid would be arginine. Okay, central dogma, B, DNA to mRNA to protein. Okay, this again is a funky one. Methionine is being carried to a ribosome by a piece of tRNA. What is the anticodon? So methionine Okay, I'm going to go backwards. Methionine, the codon for that in mRNA is AUG. Okay, I just looked at the table for that, which means the tRNA has to be complementary to that, so that would be UAC, that's the tRNA. So that's those two that I just went over, so this one and this one, they're pretty challenging. So make sure you remember when you're looking at the chart and you have your codons, those are mRNA, not tRNA. Okay, Hox genes determined in an animal's body plan. And then, for here, guys, what I did was I first transcribed it, okay, and then I translated it. And so if you find any issues with what you have, please let me know. Notice I've got GC at the end here. I can't get an amino acid based on that, so I can only make 12 amino acids, okay? Then for here, what you should know, this is a frameshift mutation. This is specifically a point mutation deletion. And so I get rid of the 5th T, so this T right here. So I end up completely, if you notice, one of my A's, this A goes away. So everything's going to be shifted. So if you look here, I just transcribed it. I didn't translate it. I'm sorry. If you notice, instead of AAG, it's AGU. So all of these amino acids afterwards are going to be messed up. And that's it, guys. So what you need to do, go back, make sure you understand everything I just explained. Go through your notes, cover stuff up, make sure you're able to write stuff down, get the names on your own, and organize your binder, and then come in and knock the socks off of this test. And that's all I have for you.